Chapter 13, Plant Growth and Development. You have already studied the organization of a flowering plant in Chapter 5. Have you ever thought about where and how the structures like roots, stems, leaves, flowers, fruits, and seeds arise, and that too in an orderly sequence? You are by now aware of the terms seed, seedling, plantlet, mature plant. You have also seen that trees continue to increase in height or girth over a period of time. However, the leaves, flowers, and fruits of the same tree not only have limited dimensions but also appear and fall periodically and sometimes repeatedly. Why does vegetative phase precede flowering in a plant? All plant organs are made up of a variety of tissues. Is there any relationship between the structure of a cell, a tissue, an organ, and the function they perform? Can the structure and the function of these be altered? All cells of a plant are descendants of the zygote. The question is then why and how do they have different structural and functional attributes? Development is the sum of two processes, growth and differentiation. To begin with, it is essential and sufficient to know that the development of a mature plant from a zygote, that is fertilized egg, follow precise and highly ordered succession of events. During this process, a complex body organization is formed that produces roots, leaves, branches, flowers, and fruits and seeds, and eventually they die. Figure 13.1. The first step in the process of plant growth is seed germination. The seed germinates when favorable conditions for growth exist in the environment. In absence of such favorable conditions, the seeds do not germinate and goes into a period of suspended growth or rest. Once favorable conditions return, the seeds resume metabolic activities and growth takes place. In this chapter, you shall also study some of the factors which govern and control these developmental processes. These factors are both intrinsic, that is internal, and extrinsic, that is external to the plant. 13.1 Growth. Growth is regarded as one of the most fundamental and conspicuous characteristic of a living being. What is growth? Growth can be defined as an irreversible permanent increase in size of an organ or its parts or even of an individual cell. Generally, growth is accompanied by metabolic processes, both anabolic and catabolic, that occur at the expense of energy. Therefore, for example, expansion of a leaf is growth. How would you describe the swelling of piece of wood when placed in water? 13.1.1 Plant growth generally is indeterminate. Plant growth is unique because plants retain the capacity for unlimited growth throughout their life. This ability of the plants is due to the presence of meristems at certain locations in their body. The cells of such meristems have the capacity to divide and self-perpetuate. The product, however, soon loses the capacity to divide, and such cells make up the plant body. This form of growth, wherein new cells are always being added to the plant body by the activity of the meristem, is called the open form of growth. What would happen if the meristems ceases to divide? Does this ever happen? In earlier classes, you have studied about the root apical meristem and the shoot apical meristem. You know that they are responsible for the primary growth of the plants and principally contribute to the elongation of the plants along their axis. You also know that in dicotyledonous plants and gymnosperms, the lateral meristems, vascular cambium, and cork cambium appear later in life. These are the meristems that cause the increase in the girth of the organs in which they are active. This is known as secondary growth of the plant. See figure 13.2. 13.1.2 Growth is measurable. Growth at a cellular level is principally a consequence of increase in the amount of protoplasm. Since increase in protoplasm is difficult to measure directly, one generally measures some quantity which is more or less proportional to it. Growth is therefore measured by a variety of parameters, some of which are increase in fresh weight, dry weight, length, area, volume, and cell number. You may find it amazing to know that one single maize root apical meristem can give rise to more than 17,500 new cells per hour, whereas cells in a watermelon may increase in size by up to 3,50,000 times. In the former, growth is expressed as increase in cell number. The latter expresses growth as increase in size of the cell. While the growth of a pollen tube is measured in terms of its length, an increase in surface area denotes the growth in a dorsiventral leaf. 13.1.3 Phases of growth. The period of growth is generally divided into three phases, namely meristematic, elongation, and maturation. Field 13.3. Let us understand this by looking at the root tips. The constantly dividing cells both at the root apex and the shoot apex represent the meristematic phase of growth. The cells in this region are rich in protoplasm, possess a large conspicuous nuclei. Their cell walls are primary in nature, thin and cellulosic with abundant plasmodesmatal connections. The cells proximal, just next away from the tip to the meristematic zone, represent the phase of elongation. Increased vacuolation, cell enlargement, and new cell wall deposition are the characteristic of the cells in this phase. Further away from the apex, that is more proximal to the phase of elongation, lies the portion of the axis which is undergoing the phase of maturation. The cells of this zone attain their maximal size in terms of wall thickening and protoplasmic modifications. Most of the tissues and cell types you have studied in earlier classes represent this phase. 13.1.4 Growth rates. The increased growth per unit time is termed as growth rate. Thus, rate of growth can be expressed mathematically. An organism or a part of the organism can produce more cells in a variety of ways. The growth rate shows an increase that may be arithmetic or geometrical. Figure 13.4. In arithmetic growth following mitotic cell division, only one daughter cell continues to divide while the other differentiates and matures. The simplest expression of arithmetic growth is exemplified by a root elongating at a constant rate. Look at figure 13.5. On plotting the length of the organ against time, a linear curve is obtained. Mathematically, it is expressed as LT is equal to L naught plus RT, where LT is a length at a time t, L naught is length at time 0, R is growth rate or elongation per unit time. Let us now see what happens in geometrical growth. In most systems, the initial growth is slow, that is lag phase, and it increases rapidly thereafter at an exponential rate, log or exponential phase. Here, both the progeny cells following mitotic cell division retain the ability to divide and continue to do so. However, with limited nutrient supply, the growth slows down, leading to a stationary phase. If we plot the parameter of growth against time, we get a typical sigmoid or S curve, figure 13.6. A sigmoid curve is a characteristic of living organism growing in a natural environment. It is typical for all cells, tissues, and organs of a plant. Can you think of more similar examples? What kind of a curve can you expect in a tree showing seasonal activities? The exponential growth can be expressed as W1 is equal to W naught e to the power RT, where W1 is final size, weight, height, number, etc. W naught is initial size at the beginning of the period. R is growth rate, T is time of growth, E is base of natural logarithms. Here R is the relative growth rate and is also the measure of the ability of the plant to produce new plant material referred to as efficiency index. Hence, the final size of W1 depends on the initial size W0. Quantitative comparisons between the growth of living system can also be made in two ways. Number one, measurement and the comparison of total growth per unit time is called the absolute growth rate. Number two, the growth of the given system per unit time expressed on a common basis, example per unit initial parameter, is called the relative growth rate. In figure 13.7, two leaves A and B are drawn that are of different sizes but shows absolute increase in area in the given time to give leaves A1 and B1. However, one of them shows much higher relative growth rate. Which one and why? 13.1.5 Conditions for growth. Why do you not try to write down what you think are necessary conditions for growth? This list may have water, oxygen, and nutrients as well as essential elements for growth. The plant cells grow in size by cell enlargement, which in turn requires water. Turgidity of cells helps in extension growth. Thus, plant growth and further development is intimately linked to the water status of the plant. Water also provides the medium for enzymatic activities needed for growth. Oxygen helps in releasing metabolic energy essential for growth activities. Nutrients, macro and micro essential elements are required by plants for the synthesis of protoplasm and act as source of energy. In addition, every plant organism has an optimum temperature range best suited for its growth. Any deviation from this range could be detrimental to its survival. Environmental signals such as light and gravity also affect certain phases or stages of growth. 13.2 Differentiation, dedifferentiation, and redifferentiation. The cells derived from root apical and shoot apical meristem and cambium differentiate and mature to perform specific functions. This act leading to maturation is termed as differentiation. During differentiation, cells undergo few to major structural changes both in their cell walls and protoplasm. For example, to form a trachary element, the cells would lose their protoplasm. They also develop a very strong elastic lignocellulosic secondary cell walls to carry water to long distances even under extreme tension. Try to correlate the various anatomical features you encounter in plants to the function they perform. Plants show another interesting phenomenon. The living differentiated cells that by now have lost the capacity to divide can regain the capacity of cell division under certain conditions. This phenomenon is termed as dedifferentiation. For example, formation of meristems, interfascicular cambium and cork cambium from fully differentiated parenchyma cell. While doing so, such meristems or tissues are able to divide and produce cells that once again lose the capacity to divide but mature to perform specific functions, that is, get redifferentiated. List some of the tissues in a woody dicotyledonous plant that are the products of redifferentiation. How would you describe a tumor? What would you call the parenchyma cells that are made to divide under controlled laboratory conditions during plant tissue culture? Recall in section 13.1.1 we have mentioned that the growth in plants is open, that is, it can be indeterminate or determinate. Now, we may say that even differentiation in plants is open because cells or tissues arising out of the same meristem have different structures at maturity. The final structure at maturity of a cell or tissue is also determined by the location of the cell within. For example, cells positioned away from root apical meristems differentiate as root cap cells, while those pushed to the periphery mature as epiderms. Can you add a few more examples of open differentiation correlating the position of a cell to its position in an organ? 13.3 Development. Development is a term that includes all changes that an organism goes through during its life cycle from germination of the seed to senescence. Diagrammatic representation of the sequence of processes which constitute the development of a cell of a higher plant is given in figure 13.8. It is also applicable to tissues or organs. Plants follow different pathway in response to environment or phases of life to form different kinds of structures. This ability is called plasticity, example heterophyly in cotton, coriander and larkspur. In such plants, the leaves of the juvenile plant are different in shape from those in mature plants. On the other hand, difference in shape of leaves produced in air and those produced in water in buttercup also represent the heterophylous development due to environment. Figure 13.9. This phenomenon of heterophyly is an example of plasticity. Thus, growth, differentiation, and development are very closely related events in the life of a plant. Broadly, development is considered as the sum of growth and differentiation. Development in plants, that is both growth and differentiation, is under the control of intrinsic and extrinsic factors. Includes both intracellular, that is genetic, or intercellular factors, that is chemicals such as plant growth regulators, while the latter includes light, temperature, water, oxygen, nutrition, etc.